Good morning, everybody.
Good morning, sir.
Before I start, I'll first share my screen to you. Can you see my screen?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Yes, any problem? Can you see my presentation?
Yes,
Yes,
sir.
Yes, sir.
Okay.
Yes, sir.
Okay. So yesterday, what we had done? Can you tell me briefly? Yesterday, I think we started with gradient gel electrophoresis. Is it correct? Dheeraj? Aishwarya?
Yes, sir.
हा आपण काय बघितलं काल?
You did have asked us the advantages of gradient electrophoresis at the last.
ओके. नाही, सुरुवात कशी झाली आपली? Did I start with gradient or did I start earlier than that?
2D, sir. 2D electrophoresis पासून चालू झालं होतं.
ओके. आयसोइलेक्ट्रिक फोकसिंग कधी झालं आपलं?
स्टार्टिंग मध्ये आयसोइलेक्ट्रिक फोकसिंग होते.
स्टार्टिंगला आयसोलेटरी फोकसिंग झालं होतं सर.
Yeah, I think I started with isoelectric focusing, and then
Yes.
I tried to cover 2D electrophoresis. Okay.
Yes, sir.
Okay. So today we'll start with something about nucleic acids. Okay, separation of nucleic acids using electrophoresis, and there is not much of a difference between the two types of electrophoresis in terms of the formats which are used. We use the same two platforms or formats, that is either vertical gel electrophoresis or horizontal gel electrophoresis, and either by using polyacrylamide or by using agarose gels. Okay, so otherwise there is not much of a difference, but the difference is here that the nucleic acids, as you know, these are negatively charged molecules, isn't it? And therefore, as we have seen earlier, in the case of proteins, what we have done is we have tried to make a buffer in such a way that the proteins carry a predominantly negative charge. Okay, so something had to be done to make the proteins negative. Now, in the case of nucleic acids, the nucleic acids are already negatively charged, and therefore there is not much we need to do about the buffers. Okay. The buffers which are usually used for nucleic acids. Nucleic acid means both DNA as well as RNA. Okay, we use buffers like tris borate buffer or tris borate containing EDTA, tris acetate EDTA buffer, or tris phosphate EDTA buffers. Okay, these are most commonly used for DNA. Now, another important thing is that DNA could be either single stranded DNA or it could be double stranded DNA. RNA is single stranded RNA, isn't it, usually? So when we are doing double stranded, when we are using double stranded molecules as the sample molecules, then there is nothing much to worry about. But if the molecules are single stranded, then there is a probability, okay, or these molecules, you can say they are vulnerable to undergoing some secondary structures. Okay, formation of secondary structures. For example, there may be some local sequences in the DNA, single stranded DNA or single stranded RNA, and these sequences may be complementary to some other sequence in the same molecule, okay, or a different molecule. And because of that, just a minute, I'll admit 3 students. Pragati, Vaishnavi, and Bharat are trying to join. Okay, so now there are 19 students in the class. Okay. So these single stranded DNA and single stranded RNA molecules, they have some sequences which are complementary to some other sequences, either within the same molecule or on a different molecule. And that is what leads to the formation of some secondary structures or some complicated structures in these molecules. Now, if such secondary structures are formed, then the problem is that they do not run very easily in the electrophoretic gels. And therefore, we have to use certain conditions which are called as denaturating conditions. Okay. So in the denaturating condition, the single stranded molecules of DNA and RNA will remain as single stranded molecules, and therefore their electrophoretic movement in the electrophoretic gels will be proper, okay, or unhindered, right? So the gels which are used for making for such electrophoresis, this gel normally contain denaturing agents like urea, for example, urea, formamide, or sometimes formaldehyde. Okay, so when we add these chemicals to the gel, then the nucleic acid molecules will remain denatured or they will remain single stranded, right? There is one more buffer which is used in nucleic acid electrophoresis, and that is called MOPS. Can you see this last sentence here? MOPS buffer, MOPS buffer.
Yes, sir.
Yes, so this is nothing but morpholino propane sulfonic acid buffer, and this is used normally for RNA, okay, RNA electrophoresis. Have you done DNA isolation in TY?
Yes, sir.
Yes, sir.
Okay, and have you done RNA isolation?
Yes,
DNA.
sir.
Yes. DNA. Yes.
Okay, not RNA.
DNA,
No.
DNA also.
Okay,
DNA and protein. Yes.
DNA and protein. Okay, so
Yes.
maybe in MSc part 2 you will probably be doing this practical, that is RNA isolation also. Okay, and at that time you will also be performing the electrophoresis for DNA as well as RNA. Okay, so coming to the next slide. Now here I have shown you three types of gel strips. Can you see the three gels? Okay, and these three gels have a difference here. One, these are all agarose gels. Firstly, the first one is a 2% agarose gel. The second one is a 4% gel, and the third one is a 5% gel. Okay, so 2%, 4%, and 5%. Now, what do you expect to happen in these three gels? 2% is a low concentration gel. Okay, so what will happen to the pore size?
The porosity will be more, sir.
Porosity increase, sir.
Pore size increase, sir.
एक थोड़ी सी गड़बड़ है बर का. There is a little bit of confusion. See, porosity and pore size, these are two different things. Try to understand me. Okay, when you say pore size is large, okay, when pore size is large, the porosity actually is less. Okay, porosity actually refers to the number of pores in a given area or in a given volume. Do you understand what I'm saying?
Yes, sir.
Yes, sir.
So these are actually contradictory things. When you say 2% gel or low concentration agarose, then the pore size will be more, but the porosity is less. Okay, and when you use a high concentration gel, the pore size will be less, but the number of pores will be more, and therefore the porosity will be more. Okay, so right now for understanding purpose, we are not going to talk about the term porosity, but we are going to talk about the pore size only. Okay, so at a low concentration, 2%, the pore size will be more. And when the pore size is more, then we can expect that the length of DNA or the size of the DNA which we can separate will be more. Is it okay?
Yes,
And
sir.
on the other hand, when the concentration of the gel is more, that is pore size is less. And when pore size is less, you can expect that the movement of large molecules will be slow. Okay, are you getting what I'm saying? I'm trying to put
Yes,
the
sir.
same
Yes,
thing in different terms, okay, different ways. Okay, so small pore size means larger molecules cannot move fast, and therefore the larger molecules will be hindered. The flow of the larger molecules will be hindered, right? Now, can you see this this phenomenon in this particular slide? Okay, who will try to explain me what you can see on the slide? Just look at
Sir,
the
in the 2%, sir, in the 2% 4% gel,
Yeah.
the 500 base pair is a very big DNA. So
Okay.
due to big pore size, it has come more forward. With respect to the 4%, the 500 base pair is in the intermediate position.
Right.
And in the 5%, it is very near to the starting point of the well,
To the
because
well. Yeah.
there the pore size is really small.
Yes. Very good answer. Very good answer.
It is unable to go forward.
हे सगळ्यांना कळालेलं आहे का? Has everybody understood this?
Yes, sir.
Yes,
What
yes, sir.
what Inamul
Yes,
has said. Okay. So if you look at the first gel here, the 500 base pair DNA has moved a little more distance. Okay. And if you compare with this with the highest concentration gel, that is 5% agarose, then the movement of the 500 base pair DNA is very slow here. Okay. It is close to the well. And here it is much far away from the well in the 2% gel. Okay. So I think you can remember this kind of image just to understand the concept about the pore size, okay, and its relation to the concentration of the gel. Is it okay? Yes.
yes, sir.
Then we come to the next slide, that is DNA sequencing gel. Now, what do you understand by this term DNA sequencing gel? It is a gel which is used for the DNA sequencing experiment. Okay, what is DNA sequencing?
Sir, Sanger's method.
Sir,
Yes, very good. Sanger's method and any other method?
no, sir, we have only done Sanger's method.
Sanger's method. Okay, the other method is called Maxam and Gilbert's method. Okay, so
Maxim Gilbert, yes.
yeah, what they do in these methods, do you know?
Sanger sequencing method. In this method, sequence of DNA.
Mhm. Yeah, that is correct. But now I would just advise everybody to, when you go home, just try to give a search, okay, and try to find out some more information about the methods for DNA sequencing. Because right now in this syllabus, although you don't have this for the exams, in the next semester, in the next year, actually you have this. Okay, just try to rebrush your memories. Okay, so what actually is happening in both the methods, Maxam Gilbert's method or Sanger's method, we carry out certain procedure, okay, on the DNA molecules, and that procedure leads to getting a set of DNA molecules, which are there is a series of molecules we get, starting from a very low molecular weight DNA to a high molecular weight DNA. Okay, we have a range of DNA molecules which are of a shorter, progressively shorter length, or you can say in other words, progressively longer length. Okay, the difference between the two DNA fragments which are very close to each other actually is just one nucleotide difference. Okay, just one base is less or more. So that is the kind of difference which is there in the set of DNA molecules in that particular set that we have. Is it okay? Now what we do is we run these DNA molecules on the gel, electrophoretic gel, for separating. What we separate, we are separating all the different types of molecules based upon the size of the molecules, right? What we are expecting is that the minimum distance between any two sequences of DNA is just one nucleotide. Okay, so when we use the DNA sequencing gel, which is nothing but polyacrylamide gel, okay, it is just polyacrylamide gel, but because of the you can say the length of the gel is very large. You can just imagine that it is about 100 centimeter long gel. Okay, how much is 100 centimeter?
1 meter.
1 meter, very good. Okay, so almost 1 meter long gel we are using for DNA sequencing purpose. Okay, the idea is that we have thousands of DNA fragments, and these DNA fragments are differing from each other by a single nucleotide in length, okay, in terms of the length. Okay, and ideally we should obtain each and every fragment on the gel. We should get a separate zone or separate band for every different size of the DNA. Okay, so for example, if you have 1,000 different molecules of different sizes, then all the 1,000 bands should be visible on the gel. Okay, and therefore the length of the gel is very large. It is 100 centimeter. Is it okay? Why the gel is longer? Why the gel is longer? Because we should be able to resolve each and every different fragment of DNA. Is it right?
Yes, sir.
बाकीच्यांना समजतंय का मी काय म्हणतोय? Okay,
Oh, sir.
so these gels are
Hello,
called,
sir.
yeah, these gels are called DNA sequencing gels. Okay, and the sensitivity of this particular technique is so good that two different DNA fragments which are differing by only one nucleotide will also form a single sharp band. Okay,
Yes, sir.
that is two different DNA molecules with one nucleotide less or more, they will form two different separate bands, sharp bands. Okay, and therefore it is called as DNA sequencing gel, right? What is the concentration of acrylamide here? Can you see? For a 80 to 1,000 nucleotide long DNA, we use 3.5% acrylamide. Okay, and if the size of the DNA is smaller, if it is 20 to 100 nucleotide, then we use a higher concentration of the gel, that is 12%. Again, the same logic. See, higher the concentration, smaller is the pore size. And because the pore size is smaller, the DNA fragment size is also smaller. Okay, so 20 to 100 nucleotides for 12%, right? And 80 to 1,000 nucleotides for low concentration of the gel, that is 3.5%, right? तुम्हाला हे सगळे आकडे जसे तसे पाठ झाले नाही तरी चालणार आहे. हे इथे पाठांतराची परीक्षा आपली नाहीये. इज इट ओके? तुम्हाला फक्त कन्सेप्ट कळला पाहिजे. स्मॉल पोर साइज, लार्जर पोर साइज, हाय कॉन्सन्ट्रेशन, लो कॉन्सन्ट्रेशन, ह्या गोष्टींचा इलेक्ट्रोफोरेसिस मध्ये कसा उपयोग होतो. दिस इज समथिंग विच यू शुड अंडरस्टँड. Okay, and then these sequencing gels are generally run in the presence of denaturing agent like urea and formamide. Why we require denaturing agents? Did I tell you earlier?
It's said to convert the double-stranded DNA into single-stranded ones.
Single.
Actually we don't have a double stranded DNA. Okay, for DNA sequencing we are using single stranded DNA molecules only, but there is a probability that the single stranded molecules will form complicated structures. Okay, if there are local complementary sites, okay, present on the DNA molecule, then because of that the DNA will form double stranded regions somewhere in the molecule, in the entire molecule, maybe at one place or maybe at many places. Okay, and when such double stranded regions are formed in the molecule, then the molecule shape will become very complicated. Okay, that is called as secondary structure of the DNA or RNA. And because of the secondary structure formation, these molecules will not run on the gel properly. Okay, if we want to run or want them to run properly on the gel, then the single stranded molecule should remain single stranded only, right? And therefore we use the denaturing agents, right? So these gels are also called denaturing gels. Okay, just remember this technical term. The gels are also called as denaturing gels. Is it okay? Well, we go to the next one. Now this is another variation of the electrophoretic technique, and that is called as pulsed field electrophoresis. Okay, pulsed field electrophoresis. हे सगळ्या गोष्टी तुम्हाला एक्झाम मध्ये येतीलच की नाही येणार हे मला पण सांगता येणार नाही. ओके, मी हे तुम्हाला सांगण्यासाठी अशासाठी हा प्रयत्न आहे की वेगळ्या वेगळ्या टेक्निक्स इलेक्ट्रोफोरेसिस मध्ये असतात हे तुम्हाला माहिती व्हायला पाहिजे, आणि प्रत्येक टेक्निकचा काही ना काही ऍप्लिकेशन आहे, पर्टिक्युलर त्याचा यूज आहे हे आपल्याला कळलं पाहिजे. हे तुम्हाला खूप डिटेलमध्ये ह्याच्या जाण्याची गरज नाही. यू डोंट हॅव टू go into lot of details of each of the technique. If you just understand the concept broadly, okay, then it is enough, right? We are just trying to understand what is different in pulse field electrophoresis and how it is going to be useful. Okay. Now this is especially useful for DNA fragments, and especially it is useful for DNA fragments which are large in size. What happens if the DNA is large? For example, if we have a whole chromosome, bacterial chromosomal DNA, for example, it's a big molecule, isn't it? The size of the DNA molecule could be, say, 1000 kb. Okay, 1000 kb is a very large size, and sometimes it is even more than that, more than 1000 kb. Okay, so when such big molecules are there, and if you want to carry out electrophoresis, then the big molecules, the DNA is like a string, isn't it? And this string is going to get coiled sometimes, coiled and intercoiled, isn't it? Because of this, that is actually leading to a secondary structure. And because of that secondary structure, the movement of the DNA in the gel will not be proper. Remember, the gel has pores, isn't it? Now, if a string is, you are trying to put a string in a काय म्हणतात त्याला, नीडल मध्ये आपण करतो ना, टाच काय सुईमध्ये आपण दोरा होतो की नाही? Are you hearing everybody?
Yes, sir.
Sir.
Yes, sir.
Yes,
Okay.
sir.
So what happens when you are trying to insert a small thread in the needle? Then what happens? There is a single pore, single hole in the needle, and therefore the job is not very difficult. You can say job is not
Sir.
very difficult. Now just imagine if there are multiple holes in the needle, and you want your string to pass through all the holes. Then is it possible? How difficult it will be?
No.
Yeah, it will be very difficult. And that is what exactly happens when the DNA molecule is large. Then it cannot go through all the pores one by one from one side of the gel to the other side of the gel. Okay. And therefore, intermittently, what happens is we have to change the direction of the DNA. Right. So in pulse field electrophoresis, what we do is we try to force the DNA molecules to go through the gel in one direction for some time. Then we stop the electric current, and the electric current will now be forced from another direction. Okay, so it's like 45-degree this side and 45-degree this side. Okay, and that is how the DNA will go on moving. Okay, through the pores of the gel until it goes to the other end of the gel, towards the other end of the gel. Are you getting what I'm saying? So if there is a chance that because of the coiling of DNA sometimes, or because of forming the random secondary structures, the DNA may get blocked somewhere. So that blockage is removed. Okay, when we force the DNA from another side or another angle, right? So what I have written here, two electric fields are applied alternately at different angles for defined time interval, say for example 60 seconds. So for 1 minute, the electric current is in one particular angle, then that current is stopped, and another electric current will be from another angle will be allowed to go. And therefore the DNA molecules will be aligned, okay, this way or that way properly, so that they enter the pores of the gel, and ultimately we succeed in the DNA, in the movement of the DNA from the wells to the other side of the gel. Is it okay? And then depending upon the size of the molecules, the DNA will get separated. So smaller molecules will, small molecules can quickly realign. If you change the direction of the electric current, then the small molecules will quickly align and they will go faster. The larger molecules will take some time. Okay. And therefore, essentially, the pulse field electrophoresis is a method for separating different sized molecules of DNA or RNA, it may be. Okay. But usually DNA, because DNA could be a large molecule. The whole chromosome of DNA can be involved in the sample, isn't it? Now, this technique is preferably used for identifying a particular disease. So it has a diagnostic value. Okay. Then restriction enzyme digested DNA of pathogens will give a characteristic pattern. Now, this is called as a fingerprint. What do you understand by this term, fingerprint? Okay. Restriction digested DNA. Okay. Restriction enzyme digested DNA of a pathogenic organism. For example, can you name any pathogen?
Staphylococcus.
Staphylococcus. Any other pathogen?
Mycobacterium.
Salmonella.
Mycobacterium. Okay, wonderful. So you know some pathogenic organisms, isn't it? So if you isolate the DNA from these organisms from a sample, for example, you do not know which DNA, which organism was causing a disease, isn't it? So from the sample, you isolate the DNA. The DNA is treated with restriction enzymes. Okay, so depending upon the restriction sites which are present in the molecule, the restriction enzyme will cause cuts in the DNA, isn't it? The DNA will get fragmented. And depending upon which DNA it is, which organism was involved, you will get a different pattern of the restriction enzyme, cleavage or digestion. Okay. So you get a certain pattern in the electrophoretic gel. What do you understand by pattern? For example, if it is staphylococcus, then you get a particular pattern. Okay. And this pattern is different from, for example, what you said, mycobacterium. Okay. So just by looking at the pattern which is found on the electrophoretic gel, you will be in a position to identify what is the pathogen. सगळ्यांना कळतंय का हे का नाही?
Yes, sir.
मी तुम्हाला व्हाईट बोर्ड वरती दाखवायचा प्रयत्न करतो. आय थिंक आय हॅव जस्ट 5 to 6 minutes फॉर दिस पर्टिक्युलर सेशन. In 2 to 3 minutes again I will start the new meeting. Okay. See, for example, I'm writing down here staph. Okay, and I'm writing here mycobacterium. Okay. Now, if I have an electrophoresis gel, suppose what I do is from the given sample, okay, I'll just try to modify this slightly. सगळ्यांना दिसतंय का हे?
हो सर.
Yes, sir.
सर
Yes, sir.
ओके.
Yes, sir.
आपल्याकडे काय हे स्टँडर्ड ऑर्गॅनिझम्स आहे. म्हणजे स्टँडर्ड डीएनए. वी हॅव अ स्टँडर्ड डीएनए taken from staphylococcus, standard DNA from mycobacterium, and then to this DNA we give restriction enzyme cuts. We treat the DNA with restriction enzyme. So what will happen when I run the electrophoresis? Suppose I get a pattern like this. Is it okay? So if I have a DNA from staphylococcus, I treat it with certain restriction enzymes, okay, and run it on the electrophoresis gel, then I get a banding pattern like this on the gel, okay. And for example, if I have mycobacterium DNA, okay, then I get a pattern like this. Is it okay? In other words, by looking at the pattern, every time I have staphylococcus with me, okay, every time I will get a pattern like this under the similar conditions, right? Every time I have mycobacterium DNA, I'm bound to get this kind of a pattern. This pattern is different from this pattern, okay? Now I have the sample from which I am isolating the DNA. Now this DNA, if I give the same kind of restriction enzyme treatment, then I'm going to get some pattern. Now, for example, if I get a pattern like this, okay, then what are you going to call this? Can you compare this pattern with the standard DNAs?
Staphylo.
Yeah. Okay.
Staphylococcus.
So this pattern, I'll just put a marking here. So this pattern and this pattern, they are identical. And that is how I can identify that the patient was suffering from a staphylococcal infection. Is it right? So,
Yes.
Yes, sir.
okay. So now I go back to my original presentation. This kind of patterning is called as fingerprinting. Okay. This is called DNA fingerprinting. तुम्ही हा शब्द ऐकलेला असेल पूर्वी. ओके, कदाचित तुम्ही याच्याबद्दल स्टडी केलेला पण असेल. ओके.
Yes, sir.
Okay, so pulse field electrophoresis can be used for DNA fingerprinting and identifying the pathogen, right? Then coming to electrophoresis of RNA. Now, I'm not going to waste much time on this particular slide. I'll just tell you something, that there are different types of RNA. You know that there is a tRNA, there is rRNA, and what is the third one?
mRNA.
mRNA. Very
mRNA.
good. Okay, so 3 types of RNA we have, and these 3 types of RNA have some distinct typical shapes, isn't it? mRNA is a linear molecule. tRNA is what is the shape of tRNA?
Clover leaf model.
Clover leaf shape.
Model.
Wonderful, wonderful. Okay, so because of these secondary structures of the RNA, doing electrophoresis of RNA is not a simple job. Okay, we have to break that secondary structure into a simple linear form, and therefore what we do is we use denaturing gel electrophoresis. Okay, again the denaturants here are formamide or formaldehyde, urea, glyoxal, or sometimes we use methyl mercuric hydrochloride. So these names are already written on the slide here. Okay, so these denaturants are used within the gel. Okay, sometimes the buffers also contain urea and formamide, and under this condition the RNA molecules are not allowed to undergo secondary structure. Is it okay? And then usually we use acridine orange for staining RNA bands. The reason is that ETBR cannot be used normally. Why ETBR cannot be used? Because ETBR is used for double stranded molecules. Okay, ETBR actually is an intercalating agent. So it will intercalate between the 2 strands of DNA or between the bases of the DNA, right? But because RNA molecules are single stranded, we use acridine orange for electrophoresis of RNA. Okay, so I think the time is almost up, and if before I start the new session, we will try to see if there are any queries from you. Have you all understood?
Yes, sir.
Yes, sir.
Yes, sir.
Yes.
Okay. कोणाला काही शंका? योगिता,
No, sir.
शुभांगी,
No, sir.
okay. Gnaneshwar, you have any question? ज्ञानेश्वर ऐकू येते का? I don't know whether he's following. शीतल जाधव. शीतल जाधव पण बोलत नाही, फक्त ऐकतीये.